One of the laboratory models that is becoming more and more popular over recent years is the zebrafish. Zebrafish are vertebrate animals that grow fast, reaching maturity in only 10 to 12 weeks. They can be housed together in tanks, with each individual fish only taking up a very small amount of space. Also, feeding the fish is not very expensive since they eat brine shrimp and paramecia that can be grown in the lab from basic supplies. At ICOB, there are several labs who study zebrafish, and one is the Chen lab. Dr. Chen often reminds his lab members about the importance of feeding and checking the animals every day, including on weekends. That way, the animals stay healthy and provide reliable research results. The zebrafish model is especially popular for studies on chemical toxicity and development. Toxicity studies are easily accomplished for a couple of reasons. First, since zebrafish live in aqueous environments, treating the animals with soluble chemicals is convenient. The chemicals can just be added to the water at different concentrations. Second, the fish larvae are very small. They can be kept in tiny containers and checked using simple microscopy. This allows researchers to test many animals very quickly in a type of experiment called high-throughput screening. For studies on development, it is possible to delete one target gene in a larva, and then the researchers can watch what happens with the animal as it grows. This kind of gene expression knockout in the animal is often accomplished by injecting the zygotic animal with custom single-guide RNAs and Cas9 protein, which is commonly called CRISPR technology. The single-guide RNAs and Cas9 work by binding the genomic DNA and cutting it. When the DNA is repaired, the target gene can be removed. The Chen lab has become a worldwide leader in using the zebrafish in new and exciting ways. We have built several new research tools using the zebrafish. With these tools, we study topics like regrowth of injured fins and development of different tissues and cell types. In some of our projects, we use fish with fluorescent protein reporters to track all of the cells of a certain kind throughout the zebrafish body. For example, our lab can now track all of the skin cells in a living and growing fish. To do this kind of experiment, our lab must be experts in building new genes, called transgenes, that can be inserted into the fish DNA and cause expression of the fluorescent protein only in the target cells. Once the transgene is created, we inject the DNA into the animal and use the animals to breed a strain of transgenic fish with fluorescent cells. In one strain, called skinbo, we have built the transgene so that each cell will have a different color. Then, we can track almost all of the skin cells at one time with our microscopy system. By building these new genetic tools for tracking all of the cells in a single fish, our lab can discover new unexpected ways in which different cell types make up an entire tissue. For more information, please refer to the description box. We hope to see you soon!